Hello, welcome and thank you for joining the second Herman Suit plenary lecture. I hope you all had a chance to watch the great legacy video of Herman Suit. He was very fond of our society and really had very strong interest in research. Some of his interests were to determine the origin of cells supporting regrowth of tumor, post-radiation, or understanding the mechanism of tumor control probability. He always had a strong interest in physics and new technologies. He was responsible for, or partially, at least in large part, responsible for getting protons to MGH and bringing proton therapy to the place they are at the moment. The lecture today is very much in the spirit of his own interests, always striving to improve cancer treatment. One of his mottos was that we have an obligation to give the patients the best treatment that we can. And so it's my great pleasure to introduce today's speaker, Marie-Catherine Vozenine, who is an associate professor at Lausanne University Hospital and the University of Lausanne. She's also an adjunct professor at UCI, University of California, Irvine. Before she arrived at this position, she studied in Paris for her bachelor, master's, PhD, and her habilitation. She had positions at the Institut de Radioprotection et de Sûreté Nucléaire. I probably messed that up. She then followed with a postdoctoral fellowship at the University of Arkansas for medical sciences and at Peter MacCallum in Melbourne. She was awarded multiple research grants, but most importantly, she also is the winner of the SIT Excellence in Mentorship Award yesterday. I think you all recognize MCAT as the face of flash radiation therapy, since she published the paper in 2014, and she will share a lot of her fascinating work with us in her talk titled From Irradiation at Ultra-High Dose Rates to Flash Radiotherapy, New Radiobiology to Support Clinical Translation. So we'll start with a short introductory video of the Herman Suit lecture and then follow with MCAT. Thank you.
Scientific research is a constantly evolving field, bringing about novel discoveries as well as opportunities to use the new information. The Radiation Research Society had the privilege and good fortune to count Herman Suit as one of its members. He was instrumental in the development of many innovations in the field of radiation oncology. Michael Goetting once referred to him as a giant of modern medical physics, summing up Herman's impact on two pillars of the radiation sciences.
You're muted.
Sorry, I'm trying to
now
share my screen. Yeah, I think I'm in. Can you see the screen? Can you hear me okay?
Yes.
Is it a full screen?
Yes. I
think we're good. Well, it's a great honor and a pleasure to be with you today for this lecture, and I must say that it's a real privilege to be giving this lecture in the name of Herman Suit. As Jan said, he pioneered many aspects of the field, and for me today, being able to tell you about the work we're doing at Lausanne University Hospital about the flash is really in this follow-up. I think it's a real privilege to be able to work in a new field where everything has to be done and rediscovered. So I will tell you today about the interrelationship in between radiobiology and the clinical translation of this field. And in fact, I will start with the clinical translation. Here are my disclosures. And as you know, today, flash radiation therapy has been really one of the hottest topics in the radiation oncology field. And we can see it with the two special issues that have been published, one in 2019 in the Red Journal, and more recently in Radiation Research. The citation numbers of these special issue related to flash are really incredibly high, showing the interest of the community in this field of ultra high dose rate irradiation. And as you say, as you know, and you've heard about that along the meeting, because now we have a lot of sessions dedicated to the flash. And again, it's very interesting, because I think that the first time I told you about this new technology was 6 years ago in Florida. I think it was my first presentation about the ultra high dose rate irradiation at a radiation research meeting. And the main interest of this new technology is really a biological effect that we've been naming the flash effect. Besides this flash effect, some other interest benefit of this technology are visible, like motion management, like treatment time and cost. But I will really focus about this biological effect. And this biological effect involves two components. On one hand, normal tissue sparing at dose that normally cause normal tissue toxicity when we use conventional dose rate, and on the other hand, are able to equally eradicate a tumor as compared with conventional irradiation at conventional dose rate. So this is really a two-aspect definition, and I really want to stress out here that it is a definition that has been developed in vivo. And here I want to be clear, because I saw previously in the meeting quite a lot of talks and experiments related to in vitro experiment. For now, this difference in between normal tissue sparing, this differential effect in between normal tissue sparing and tumor control has to be performed in vivo. There is no in vitro system that is able today to achieve this this goal and to be able to objectivate this differential effect. And I want to tell you proof of that, showing that in fact, when you look at the clonogenic assay, which are the gold standard in our field, you don't see an eradication of the tumor when you modify the oxygen concentration. You really see the reverse. So for me, if you want to define the flash effect, you have to work in vivo in a tissue component. And as you see, there has been a lot of publication in this field by my group here in blue. And I will also show you that there has been a lot of publication by many other groups in green, using electron beam, using proton beam, a little bit with a photon beam, and some negative results also with photon beam, proton beam, and electron. And I think it's very important to understand the difference in between all those results and data. I think this slide cannot become more busy, and since the number of publications that have been produced and that will be produced in the future in this field will become higher, I won't be able to show you a global overview like that any longer in the future. So as I said, I will start with one big question that we are asking ourselves is how to use this flash effect and the opening of the differential effect in between normal tissue and tumors in the clinic. And I think there is 3 big questions that needs to be investigated and answered: the technology, the impact of fractionation, the impact of volume is also a very important thing. And for today, the application for the clinical translation are very limited by the device we have and that are able to operate at ultra high dose rate. At the CHUV, we are using intermediate energy electron that have been nicely summarized here by this in this review by Wilson. The proton field is also expanding the application, and the X-ray field is still moving on, and there is still no X-ray application feasible in the clinical setting. But in any case, for all those applications, we are using what is currently technologically available. And what we really need, and what I really want to stress out, is that if we want to move forward, we really need to improve this from the technological point of view, think outside the box in order to develop new machines, in order to be able to perform irradiation at ultra high dose rate in patients. And here I show you an example of this possible development with photon accelerator at ultra high dose rate by a Chinese group. This paper has still not been peer reviewed, but it's probably a way that can be a topic that can be expanded. Now, I already showed you this slide last year, and it's about parametrization. And you've seen that Pierre Montegruel and Jean-François Germont in the group were very active in order to plot together the conditions that would be required to obtain or miss the flash effect. And as I told you last year, this parametrization gave rise to the impression that we had very important parameters that are the dose rate within the pulse and the irradiation time for the delivery of a given dose. And from what I've heard in the meeting, it seems that everyone was quoting our first dose rate, the first dose rate we've been using with Vincent in the first publication in Science Translational Medicine, which was 40 gray per second. But is 40 gray per second the magical dose rate that we need all to use and to use as a reference? In fact, I don't think so. When you look at this plot, you see that probably being higher in the dose rate and lower in the time will enable us to really get out of this gray zone that I show you here in the middle of the plot with all the cross that are negative data, and will probably help us to move forward. What we think for now, when we have been compiling all the recent data, and you see here that Jean-François has included the recent publication by the different group, we think that in fact the dose rate in the pulse, the higher the dose rate in the pulse is, the better it would be. The shorter the time is, the higher it would be. And it is very well supported also by this recent paper by coming from Christopher's group in Oxford, showing that the temporal structure of the beam was really, really important, with the flash effect being lost here in this model of crypt survival in the gut, when the delivery time was increased. And it was also lost when the time in between the interval of the pulses were increased. So it means that we need to investigate those questions, and we also need to investigate this question related to what we have in the proton field, because the proton beam currently are working in a very, very different way as compared with the electron beams that we've been using a lot. I told you about the importance of fractionation, and there is not so many studies investigating fractionation. With Pierre, we've been able to show that fractionation was feasible, and Marc Mendonca did a very nice work in summarizing and making a very nice cartoon to show that indeed fraction size matter as well as the total dose in order to keep the differential effect that we wanna achieve. One also very fundamental question that has not been addressed a lot so far, because most of the data were produced in mice, is what is really happening in humans, in human cell. And this is one of the few results that have been performed using human cells, and in that case, human PDX, with my colleague in Paris, Françoise Plumiot and Benjamin Huzon. I think I already showed you this slide last year. And doing this experiment, we showed that in fact, not all tumor cell lines were equally sensitive to flash irradiation, with this one being pretty much resistant to flash irradiation. And when we were looking at the reason why it could be, we defined a putative gene profile involved in this susceptibility profile in the two susceptible lines. And one of the very relevant genes that came out from this analysis was the gene GAD45. So we decided to go a little bit further, and you heard that Ron has been doing a lot of RNA-seq investigation. And from those RNA-seq investigation he did, in addition to the work he's been performing with the oxygen, and we will talk about that a little bit later, he has also been able to show that the major hallmark of the G2M checkpoints, as well as mitotic spindle group of genes, were altered after flash irradiation, with especially relevant upregulation of genes related to PLK1, polo-like kinase 1, as well as CDK1. And this supported the relevance of the G2M checkpoints in the response to flash irradiation. And it's now investigated in the lab by Emric Almeida. And Emric is using different type of inhibitors in order to work to block the cells in the G2 phase of the cell cycle. And very interestingly, when it does that and blocks the cells with this inhibitor, which is called RO3306, which is a classical type of inhibitor, it is able to increase the number of micronuclei 72 hours post-irradiation, specifically in the flash irradiated group, whereas this impact in the conventional irradiated group is very, very low. So it's really support the idea that this modulation of the G2M phase will be of real interest to investigate in order to understand the sensitivity of tumor cells to flash irradiation. Now, we were still in this clinical part, and I would like to tell you a little bit about what has been done, or what is currently being done in human patients. So you all have seen this work published by Jean Bourris and done at the CHUV, treating a first patient with our experimental beam ERT6. As I said already, because I already showed you this work, it was more a feasibility study in order to know how we could work with the official agencies here in Switzerland, and it was really feasible to work with them. In the meantime, Jean Bourris has started another clinical trial with the machine from IntraOps. This is called the Impulse trial. It's targeted to the treatment of superficial tumor because again, we are using very intermediate energy electron beam. And in the meantime, the group of Cincinnati Children's Hospital, along with Variant, has launched the FAST-01 clinical trial in using the proton beams that they have validated earlier at the preclinical level, both at the normal tissue and the tumor level. And you see the publication here, quoted here. And in this FAST-01 trial, they are treated symptomatic bone metastasis in a very careful way with a dose of 8 gray. It's really also to see whether it was feasible to implement this type of approach at the clinical level and work also with the regulatory agency and try to fit things together. But in any case, and I will show you why, more investigation are really needed for a safe clinical transfer. And in fact, we are today at a very specific time point of the development of this technology. We are at kind of a switch where we can move forward in a safe way in order to be very beneficial for the patient and for the community, or we can really do harmful things if especially the safety is not taken into account. And really this is in the hand of the radiation oncologist now to be able to do a safe transfer. I think we still need some preclinical investigation, and I'm going to show you why. The first point is most of the data that have been published so far with any beams that we've been using, electron, proton, photon, have been using mice, very small animal and very small volume, either single dose or fractionated, hypofractionated type of treatment. But if we want to go and treat the patient, we're going to need to be able to treat a large volume. When you ask to the proton people how they're going to achieve that, the answer that they can do it with the pencil beam scanning, which still ask a big question about the time. How are you going to be able to achieve a large volume in PBS in the time frame needed to achieve the flash effect? I think this has to be investigated in a very rigorous way. So if we look at this question of the volume, and I show you the data we've been obtaining with our peak a model. Here we have been comparing a low a small volume of irradiation, a single spot of 2.6 centimeter that has been irradiated with increasing dose of irradiation, and you have here the parameter of the beam. You all have seen those images. They are published. This is the conventional site with the fibronecrosis 9 months post-irradiation, whereas the flash irradiated site is nearly normal. This is sustainable in time with dramatic retraction of the tissue 3 years post-irradiation in the conventionally irradiated spot, whereas the flash is still very normal, a little bit of hyperkeratosis. Now, if we want to transpose this irradiation to a larger volume, the story is very different. Here we have not been able to get the authorization from the Swiss authority to do the conventional irradiation in parallel, and you understand why, given this large fibronecrotic spot that we obtained already with the small volume. But when we treated this large field of 8 by 8 with flash parameters, you can see that the story is pretty different. With development of telangiectasia, 4 months post-irradiation translating into ulceration and fibronecrosis, 5 to 6 months post-irradiation with the retractation of the tissue, and complete healing after 7 months, translating into a scar, which is in fact, now at the histological level, characterized as an atrophic type of remodeling in the skin of the pig. So if we move our parameters, we have been forced to degrade a little bit those parameters to achieve this large field of irradiation, and that's why we also need to work further in order to develop the accelerators to do those type of experiment. You see that here, instead of using 10 pulses, we've been using 20 pulses, and I told you from the work done by Christopher and by Pierre before that, in fact, we know that our best parameter are when we are delivering only 1 pulse, but we cannot achieve this 1 pulse with high dose rate and this large volume. So we degraded our parameter of irradiation, and we end up with toxicity at the normal tissue level. We have been also moving forward with study with domestic animals, and especially cat patients bearing spontaneous squamous cell carcinoma. These studies are done in collaboration with Carla Rohrer from the Zurich Vet School, and it was done in the frame of a phase 3 clinical trial. I show you here the 2 arms, which is the standard of care for this trial, and here the second arm is flash. In fact, probably the original slide showed you a dose of 34 gray because this dose was the prescription interface, but here we were homogenizing the expression of the dose delivery to be in the isodose 90 as compared with the standard of care. So we've been delivering 30 gray in a single fraction, 3 pulses and 20 milliseconds. So here, very small volume, 2.6 centimeter, and a dose rate very much in the best condition possible. But I can tell you already that this trial has been interrupted, and it has been interrupted after 13 months because of this cat, Moussi. And this cat, Moussi, in fact, developed, as you can see here in this picture, an osteoradionecrosis. And this occurred in 3 of the 7 cats that we've been treating. And with Stillbollen in the group, we've been performing some reconstruction studies in order to try to understand what was happening, until found that in fact we had some hotspots of dose deposition with an additional 125% more dose at the level of this hotspot. TIL is currently doing some more reconstruction in the 4 cats without osteoradionecrosis of the maxilla. But it means that here, this 30 gray given in a single fraction, despite the fact that we were with a small volume, and despite the fact that we were in a very, very high dose rate condition, is triggering toxicity. So again, and I think I already told it last year, flash is not magic. If we are pushing the system, we're going to increase the toxicity, and we're going to see toxicity appearing. And we are not the only one doing some clinical trial with pet animals that are wonderful model. I see that there will be a specific session after my talk. Mainly for now, what has been published is short-term investigation. And as I just told you, our results show that late toxicity investigation are needed. And it's kind of opening open door in the radiation oncology field. What is worrisome and what we need to take care about is really late toxicity. So we need to be clear-cut and be able to characterize those in order to move forward. So what remains to be explored in order to be safe? I think one big question we have to ask is, does one dose rate will fit all purpose? And I'm afraid that the answer is no. I saw also in many occasions during the meeting that a dose of 10 gray single fraction was quoted all the time. But it does not mean that fractionation is not feasible. And it does not mean that conventional fractionation is not feasible. It just has simply not been done. So we need to conduct those fractionation with conventional type of fractionation, fraction of 2 grays, in order to know whether we could be able, when we use ultra high dose rate, an additional fraction, 2, 3, 4, 5, up to I don't know how many. It's going to be very tedious type of investigation, but it's going to be needed. We also need to investigate the impact of dose rate versus volume versus organ. And as I said before, the effect in human is very important. And here I think that the organoids and organ on the ship are really a place to play, a role to play. There is another important thing that needs to be investigated, is the combination of flash with other type of therapies that are the standard of care in radiation oncology. And that's why I really want to stress out. I think this paper published by the NCI group point really the relevant question about the technology and more biological investigation in order to be in the safe side for the development. Now, let's go back a little bit at the mechanistic level. And at the mechanistic level, I think that here as well, we really need to be able to think outside of the box. And we really need to re-explore the radiation biology in order to find explanation for this impact of ultra high dose rate irradiation. I think that really using standard radiobiology is going to be something that's going to be challenged in the next future. And I want to show you a little bit the strategy we've been using in Lausanne. And we are really lucky because we are a really multidisciplinary team with physicists, with chemists, with biologists, with physiologists and clinicians. So we have a kind of an integrated system. And when we started this work, we in fact it started with the biological step, and we investigated what was happening at the functional level in the brain. And this was done in collaboration with our colleagues from UCI, Charlie Limoli team, and you have all the team here, along with the biologists at CHUV at the time, Pierre, Jonathan, and Benoit. And with the help of Charlie team and Munjal Acharya, we've been using a lot of different functional endpoints in order to investigate functionally the impact of this flashy radiation at the brain level. And using all those different tests that the group of Charlie has developed, we've been able to show that flash was preserving the cognitive skills of the mice, and they were always equivalent to the one of the control mice, as if they were not seeing the beam, in fact, when compared to the conventional part. And Imric, a young PhD student in the lab, made a very nice summary slide of what we have found at the brain level when you look both at tumor control as well as normal tissue impact when you irradiate at conventional dose rate, 0.1 gray per second, and flash above 100 gray per second. And whereas always we found iso efficacy at the tumor level, with flash we had less neuroinflammation, whereas with conventional we always found microglial activation and astrogliosis. You heard that from Pierre on Monday. We were able to maintain the vascular integrity, whereas it was always disrupted with conventional irradiation. And you saw that in Barrett Allen paper last year. And as I said, we also always showed neurocognitive sparing. And one of the hypotheses that was that it could be dependent upon the production of reactive oxygen species after flash irradiation and perhaps depletion of oxygen. And I will go quickly over this hypothesis because you already heard a lot about this hypothesis in the past few days. But our idea was coming from this experiment done by Pierre when he delivered carbogen to the mice and was able to reverse the neurocognitive sparing effect of flash irradiation. So it provided us with a testable hypothesis, which is very interesting. When you have something to test, you can really design some experiment in order to test the hypothesis. And today we know that this hypothesis of oxygen depletion is wrong. It's not true. The measurement done directly with some probe do not support any radiolytic oxygen depletion at the therapeutic dose 10 gray that we have been using in our whole brain irradiation experiment, both at the tumor and at the normal tissue level. And this has been nicely published and confirmed by the group of Sergei Vinogradov and Brian Pogge. And you have heard about this last year by Xu himself at the radiation research meeting. Flash RT resulting in insignificant oxygen depletion. It has also been confirmed by another type of probe. Here we have we are speaking about 3 different probes. Velko Grell in the lab used the Oxylite. Pogge's team used the Oxy4 probe developed by Sergei Vinogradov, and the group of Heidelberg used another probe, and all of them are ending up with the same conclusion. But as you know, when we are doing such experiment, and when we really want to prove an hypothesis, only one type of measurement is not sufficient. So we went on further, and we decided with a young chemist in the lab, Uda Kassem, who has been trained by Gérard Balaccino in Paris in radiochemistry. We decided to go and look really at the initial production of these reactive oxygen species. And what Uda did was to evaluate the primary radiolytic yield, so the G0 value, using a very well-known and standardized method which is using scavengers. So it is a chemical system that has been developed in order really to come back to the very, very, very early event of the production of this reactive oxygen species. And here I show you the G0 value she found for H2O2, both conventional and flash dose rate with our Oriatron ORT6 at the CHUV, and you see that they are not different, and they are also very consistent with the previous report performed with a different type of rays, cesium as well as cobalt. So from this data, it really supports the fact that we don't have any radiolytic depletion at the initial level. But again, we wanted to be really sure, because Uda was working in a very simple system with water, and you never know what's going to be happening if you are using more complex system. So Nicolas used a very classical type of method and came closer from the biological system. He used plasmid irradiation in order to investigate the damages, single strand and double strand damages at the plasmid level after flash and conventional irradiation. So I won't explain to you what this assay is, as it's very classical in the field. So it did some dose escalation, look at the different type of plasmid forms that show single and double strand break, and he irradiated the plasmid either in atmospheric condition or in physioxic condition. And you can see whatever it is, single strand or double strand, the flash and the conventional curve overlap, and it's true at atmospheric oxygen condition as well as physioxic oxygen condition, 4%. So consistently with what we have been obtaining with a very simple water model done by Uda, the results obtained by Nicolas with the plasmid shows that the damages at this level are very similar in between conventional and flash irradiation, and those data are currently transferred to Sébastien Certi and Laurent Desorgueur in order to implement them in the Géant4 DNA model. But we still have these curves that have been reproduced quite a lot. And when you have been looking at later time point in the cascade, after the diffusion phase, in fact, here you have a difference in between flash and conventional irradiation. And it's still feasible even at the lowest dose of 5 and 10 gray, meaning that in the diffusion phase, there might be a difference in the level of reactive oxygen species in between flash and conventional. Whether this small difference has a significance at the biological level is a question. We are working further now with UDA using more biological model, zebrafish system, as well as cellular system in order to investigate this question. We are also working with a group of duck spits in order to move further into that that question, but this is still ongoing. In any case, what we can say from what Ron Lewitt told you on Monday is that the relationship in between oxygen, hypoxia, and flash efficacy is a protection of cytotoxic effect. It's much more complex because from the model that have been published recently, we thought that hypoxic niche would be sparing the cells, whereas, as he showed you, it's not the case at all when you are using a very artificial clamping system where you have a very severe hypoxia. So if we want to summarize a little bit what we've been doing in cell-free system, we don't think that radiolytic oxygen depletion has anything to do with the flash effect at the therapeutic dose that we use in the range of 10 gray. The H2O2 primary yield is similar in between the two modalities. There is a little bit less production of reactive oxygen species, in that case H2O2 with flash, but does this as a real biological significance is really in question. So if it's not about reactive oxygen species, what else could it be? And if you look at what we can have in a cell, there is some other possible mechanism related to protein oxidation, related to oxidative alteration of the mitochondria, and you remember that the electron transport chain in the mitochondria is relevant. Those questions are investigated, and especially with Doug Spitz's group at Iowa. But I want to point something out a little bit more today, which is lipid peroxidation, which was an idea that was brought into group by our chemist, Pascal Froidevaux, who was really interested in this type of lipid peroxidation, which is highly relevant in biological tissue because of the membrane. And we also know that this type of system is limited by the oxygen availability. So we are still back with our oxygen concentration. And I'm going to show you the impressive results, in my opinion, that Pascal has been able to obtain using HPLC and measuring MDA production in cell-free system. So he used either micelles of linoleic acid at the top or liposome composed of phosphatidylcholine at the bottom. And you see the major impact, differential effect in between conventional in blue and flash in red, related to the production of MDA which is an outcome of lipid peroxidation in this system. So here, I must say that it's the first time we find something that different in our investigation, and this is really thanks to Pascal. And it brings me to this intermediate conclusion. Here we are in cell-free system. What about in cellular situation? It's our next step of investigation. And I'm nearly done, but I really would like to go a little bit to more complex biological system and tell you a little bit about the benefit of hypofractionated flash irradiation in a model of adult and pediatric mice. And this is a work that is mainly done with by Yaz Alakban at UCI. We are irradiating the animals and send them to UCI. Yaz and Charlie team are investigating the impact of this irradiation on adult mice and pediatric mice. You see that we have used different protocol in adults, 3 times 10 gray, in pediatric animals irradiated at 3 weeks of age, 2 times 10 gray, and Yaz performed a huge amount of behavioral tests that they are doing at UCI, and here she has been also able to have access to the response of males and females, and you can see that in most of the tests she's been doing, she has shown a very nice improvement in the adults after fractionated irradiation. So very nice sparing of the cognitive capabilities of this animal, and it was also the same in the pediatric, but mainly in males. So there is probably something very important to investigate there in between males and females in order to move forward. And of course, the hormonal status is of a huge interest. And if we are going back to the lipids, perhaps we're going to be able to make a loop. This is the last piece of data that I want to share with you, and you have already seen it partially shown by Pierre on Monday. What is really striking in this experiment done to investigate the synaptic plasticity using a very specific type of assay, which is called LTP, and which is done by neuroscientists at UCI, the group of Marcello Hood, with collaborating with Charlie's team. We've been able to show that flash irradiated animals behave really like the control animals, whereas we had an alteration in the conventionally irradiated animal. And it's true in the adults, and it's true in the pediatric animals, means that this factor is probably a very potent trigger for the preservation of the cognitive function in our animal after flash irradiation. And I want to end up with this last piece of data that is coming from the group of UPenn and is to me very, very interesting and very, very relevant, because it goes into this description that, in fact, at the normal tissue level, flash irradiation is not doing something. It's more conventional irradiation, which is activating a bunch of pathogenic signal, including various genes that you can see here in this plot, and they are not activated by flash irradiation. Another piece of data that Costa showed us yesterday is very intriguing, is the response, is the investigation he has been doing in association with David Kirsch with the P53 deficient animal, who showed nearly no impact or a very, very delayed impact in the survival. Means that really we have a major biological difference to investigate. And with that, I would like to end up this talk, and share with you one thought. We've been thinking a lot about ultra high dose rate irradiation to promote flash radiation therapy, and it's a wonderful goal. But what about if we were having a tool in hands, a unique tool in hands, to explore the fundamental difference in between normal cells and tumor, something that we were not seeing before, and that ultra high dose rate irradiation and work with ultra high dose rate irradiation can help us to decipher. So with that, I'm gonna thank you, and I would like to welcome you all to the FRPT meeting that's going to be held in Vienna. It's going to be in person, so hopefully we're going to be able to meet each other. Come and join us. And I really want to end up this talk with thanking my Flash Dream team in Lausanne. You see us in our ski gears with the wonderful mountains behind us. This is the biology team with a bunch of young and very talented students, postdocs, collaborators. So it's a great privilege to be able to work with them. I really also want to thank the physics team. Without them, it won't be possible to work. So it's the team of François Boschu. Here, I really want to highlight the contribution of Pascal Froidevaux. We have also a very, very strong clinical team. I want to thank our collaborator, but I cited them along the talk, as well as our funding agencies. And I thank you for your attention. I will be happy to take any question.
Thank you very much, Kat. That was, as always, I'm blown away by all the amount of data that you present. You think you know what's going on in Flash, but it's always more than there is, and the story obviously is still far from over. I would like to use this as a short plug. This was a really great Herman Soot lecture, and we still need a little bit more to fully endow this lecture. So if you are willing, please consider giving to the Herman Soot Lecture Fund and the RRS website. And we have a few minutes left for questions. Sadly, I think we need another half an hour for that, but let's start with the first one here. From Rich B, the emergence of the late effects in the cat study are very concerning. These observations are also bring into question the cognitive sparing that have been reported. If these were done at a later time point, would you see sparing?
Yeah, we've been doing that. We've been investigating the sparing 2, 6, 9 months post-irradiation in the mice, and I can tell you that it's stable. I also can tell you that we've been performing similar experiments with old animals, and we are also able to keep the cognitive sparing. So in the mice, again, we are using a very small volume and 1 pulse. So it means a dose rate 10 to the 7 gray per second, which is not what we could use here in the cats. Plus, we increase the volume, and what is known is that the volume has a major impact.
Right. Let's try to get to some more. Was the plasmid work by Nicolas done in high scavenging environment?
No, it was done in water.
Okay, I'm sure we can also read up on the paper for all the details.
Well, it's not published for now.
When it's published. Or we find him at the flash radiation workshop in Vienna.
Yeah, for sure. You will.
All right, we'll go down here by, are the levels of lipid peroxidation different from tissue to tissues?
I don't know. We have not done that for now.
Right.
No. Well, yeah, it's a great question, and for sure it's going to be the next step of the investigation for sure.
It seems that putting together data showing that increasing oxygen levels can impact the therapeutic ratio. Is there a role for altering oxygen levels in tumors together with FLASH?
Yeah, but then you're going to lose the differential effect. So I don't see really the point.
Maybe altering oxygen levels in normal tissue?
Yeah, but then you lose the normal tissue protection. Now, of course, you're going to increase the tumor toxicity. Well, I would not go that way, but up to you. Any idea is welcome.
Yes, we should study it all. All right. We have some more minutes. Fascinating observation regarding the late oxidative stress, uncoupling from mitochondrial oxidative phosphorylation. Some evidence that this is one impact of standard radiation exposure. The mitochondrial.
Well, I think that the next step of investigation is going to be there about the redox metabolism, and as I said, we have a collaboration with Doug Spitz's group, and hopefully we're going to be able to come up with some answers very soon.
Okay, so we have to go over a few minutes, so let's continue with the questions. One question from Anonymous: could the FLASH effect relate to RNA more than DNA?
I don't know.
Okay.
I don't know. I never asked this question to myself. So what do you mean really about splicing or stuff like that?
Maybe messenger RNA or so, but
well, gene expression, yes, but yeah, I have trouble to understand this question, but perhaps you can send me a message and we can exchange.
From Reza, what is the minimum dose to observe FLASH effect? Is it tissue dependent?
In fact, it's exactly what I said. For now, we have been working mainly with high doses, okay? 7 gray was the one that was published in the hypofractionation study, because we need to have the toxicity at conventional level, okay, to see the difference. But it does not mean that we cannot observe it after 2 gray fraction. We have to do the experiment before we make this conclusion, and it has never been done so far. So there is, for now, there is no minimal dose really investigated. It has been done with high dose, 7 gray and above. But let's see. Let's do the experiment before drawing any conclusions.
From Pierre, what a fantastic talk. Do you think that hypofractionation could prevent the osteoradionecrosis in cats?
It's going to be, it's going to be the next step, of course. When we're going to do, we are planning a next trial with Carla, and we're going to do that. We're going to do hypofrac. Yeah.
All right. Impressive pioneering work. Thank you for this excellent presentation. By the way, if anyone wants to go to the other sessions, please feel free to leave. I know we're a little bit late here. Impressive pioneering work. Thank you for this excellent presentation. Can you comment on how immune response can fit into the FLASH effect? What is your opinion about potential benefit of FLASH for anti-tumor immune response?
Well, what we know is that at the normal tissue, FLASH does not activate the immunosuppressive impact. So the TGF-beta is not activated. From the data that have been published so far at the tumor level, the curve really overlap. And if you looked very carefully in the work published by Mathieu Sartorio group, they did experiment with immunoactive and immunoindolent cells, and they overlap. So I'm sorry to tell that I don't think the immune component has too much to do with the FLASH effect. But we will come with some data, and it's going to be presented in Vienna by Emmerich.
I'm looking forward to Vienna. From Manuel, thanks, MCAT, always amazing. Is there any experimental plan to disentangle volume effect and dose rate parameters?
Yeah, it has to be done. Huh? I don't think I will do that. It's very difficult for us in Switzerland from the ethics point of view. So if anyone want to jump in, please do. we need it, but we're gonna need to make that with big animals.
Let's see, another one for lipid peroxidation. Great talk from Kunal. Great talk. Thank you for the lipid peroxidation observation. I'm interested in. He will be performing more experiments. Great, please do. Any age effects or gender effects with FLASH? I mean,
gender effect, yes.
Yes.
And no difference in pediatric, adult, and old. Well, I mean, no difference. The FLASH effect can be seen. The sparing efficacy of ultra-high-dose irradiation can be seen in all this situation. The old animals were 50 weeks plus. We're going to publish that soon, hopefully.
Okay. Have you looked at mitochondrial fitness or toxicity in long term after FLASH with conventional on the lipid peroxidation results?
It's ongoing. It's ongoing. We don't have any data on that for now.
I think I've seen a lot of data this time that long-term effects really are so we have to study things much longer than we think we usually do.
Yeah.
How many mice can be irradiated at once, and how many mice were in each FLASH group in the mouse experiments? Is it one mouse at a time?
You're talking about the cognitive impact. We have a group of 16 animals in any condition, so it's a minimal of 16 animals in each group.
But one mouse at a time irradiated.
Oh yeah. One mouse at a time. Yeah.
I think that's for most cases, most experiments.
Yeah, and always a dosimetric backup for each mouse.
One more, the radiation-induced membrane, ceramide-dependent microvasculature endothelial cell death was shown important to contribute to the epithelial cell compartment integrity. A role here for FLASH?
Good question. No answer. You see, it's a completely open field, so that's just wonderful. We have a lot to do.
That is a great way to end it. There's still lots to do, so please do your research. Do join the
do experiment. Stop modeling.
Hey,
that's for you. That's for you, Jan. A little bit, a little teaser.
Right, I'll try to do both. All right, thank you, everyone. See you in the next sub. Bye.